Thank you so much. I'd like to take the opportunity to thank the organizers. And I'm really sorry I couldn't make it in person. It happens to be a favorite city that I keep visiting, but this time I just couldn't cut it. So I've been asked to speak about newer antibody drug conjugates in breast cancer. So coming to it, I'd like to go a little specific into what it is. This is basically a monoclonal antibody, and it's linked to a cytotoxic drug. So what you do essentially is that you are actually limiting the delivery of the active agent to very few cells. That widens the therapeutic index. Uh, Theoretically, what it should be doing is it should be kind of allowing us to give a higher dose. So that is how you widen the therapeutic dose there. It has three components to it. You have an antibody, you have a linker, and a payload. All these three are very important for how a ADC functions. Now, if we look at the antibodies, antibodies can be IgG1, G2, G3, or G4. The basic difference is what is the serum half-life and what is the avidity with the F fraction. Now, if we look at G1 and G2 and G4, they have a very long serum half-life for around 21 days. If we look at what is the F fraction binding capacity, it is the maximum for IgG3, but usually the serum half-life is somewhere around 7 days. So most of the times we end up using IgG1 as our this thing. Linkers, what we need to understand is that there are two categories majorly. One is a cleavable and a non-cleavable. Now, in the last speaker has made my job quite easy when he spoke about the bystander effect. And this is the part which tells us about the bystander effect is what is the linker. If the linker is cleavable, that means that it will cleave the active agent will be broken off. And after it has broken off, it is free to spread into the tumor microenvironment. Second problem with a cleavable would be that once it is cleaved, it can get cleaved even while in circulation. So your side effects might be a little more. The payloads, you can either have antimicrotubule, you have DNA cleavage, you have topoisomerase inhibition, which is actually the one which will be also working. Now, if we look at what is the mechanism of actions of ADCs. Now, when we have ADCs, these ADCs mostly circulate as three components. Some of the payload might not actually be attached to the very antibody. Some antibodies will be free. Most of them, you get up it's a composite molecule. That is how you get the drug antibody ratio of 3.5, 7.7, 7.4. It is because some of the preload is not probably gets dissociated when it is in circulation. As they slowly diffuse towards their targets, the ADCs can release some payloads into the tumor microenvironment, but most of it tends to attach to the directed antigen. There are two aspects to it. Let us take, for example, trastuzumab deruxtecan. Now, in trastuzumab deruxtecan, the trastuzumab component is still effective. The trastuzumab per se, after attaching, is able to get the NK cells and per se can have a payload independent anti-tumor mechanism. Second, it could be, let us say, once, but most of it is internalized. Once it is internalized in the lysosomes, the linker is cleaved. Now, once this linker is cleaved, this payload or the molecule which is actually that we have attached to it gets into the cell, and that is where it has its action, either whether it is on acting on the microtubules or it is going on to act on the DNA, depending upon what is the payload. Subsequently, these cells, these payloads may diffuse out. Once the cell is lysed, it may diffuse out. It can have cells which have not directly been attached by the antibody conjugate, but because the payload is out in the microenvironment, they can have something known as a bystander effect. Now, this is one of the few differences that has been brought out by the previous speaker also between TDM1, which has a kind of a non-cleavable linker, and deruxtecan, which has a kind of a cleavable. Now, coming to it, the ADCs in oncology started quite early. 2001, we had the first approval, and we had gemtuzumab ozogamicin. It was later recalled, but subsequently, that is what paved the way. Then we had brentuximab vedotin. For the breast cancer, coming to it, TDM1, the first approval was in 2013, followed by deruxtecan and subsequently by sacituzumab govitecan in 2020. Now, if we look at the FDA approved ADCs that we have TDM1, trastuzumab deruxtecan, and sacituzumab govitecan. These are the three. So probably I'll feature first spend a little more time on these, then subsequently go on to the newer ones. So if we look at trastuzumab, it is a IgG1 antibody. It's got a non-cleavable linker, and it is linked to mptancin, which is a microtubule inhibitor. If you look at the drug to antibody ratio, it is 3.5. So for every molecule of trastuzumab, you have roughly around 3.5 molecules of mptancin which are attached to it. Now, all of you are aware of this trial. We have spoken about it in earlier this thing, so I'll not be spending much time on it. This was, as you know, lapatinib, capecitabine versus TDM1. The subsequently, this is something not in metastatic settings, but in Catherine trial. This is in the residual, no pathological response. Here again, we had a kind of TDM1 did better than trastuzumab. But coming to the next drug, the trastuzumab deruxtecan. Now this again, if you look at it, it's got a IgG1 antibody. It's got a cleavable linker, exatecan derivative, which is actually topoisomerase 1 inhibitor. And you look at the drug antibody ratio. It's about 7.7 or 8 molecules of the exatecan derivative to each molecule of trastuzumab. Now, this was the preclinical data that we had that was published. This was in somewhere around 2016. Now, and subsequently, the bystander effect was subsequently again clarified. First thing, it retains activity of trastuzumab. So trastuzumab component per se also acts. It inhibits the topoisomerase 2 and has a bystander effect. Now, this is what the recent Destiny Breast 04 is taking care has explored what was initially seen in preclinical data, that when you had a co-culture of HER2 positive and HER2 negative cells, TDX kills both HER2 positive and HER2 negative breast cancer cells. So you just require this molecule to reach up to the cancer cells which actually have got HER2 new on them. So whether they have it in a homogeneously large, whether it is HER2 new 3 plus, 2 plus, or 1 plus, once it reaches there, it gets cleaved, and you, because of the bystander effect, you are likely to get the clinical results. And subsequently, there were these monkey studies on the monkeys, which again proved that the TDXD was effective in animals who had previously been exposed to TDM1. This, of course, was translated into clinical trial. Initially, they were looking at the dose phase 1, phase 2 trial, wherein 5.4, 6.4, and 7.4 was compared. Now, what was to be seen is as you increase the dose, the effect increased, but the amount of side effects also increased. And that is why they got a cutoff of 5.4 milligram per kg. It had 6% CRs and such a beautiful waterfall. This tells you that you have a very, very effective drug on your hands. And as you can look, 54.9 PR rate. So this gives you about somewhere around 61% clinical response rate. And in this heavily pre-treated, you had a median PFS of 16.4 months. Safety, yes, this has been spoken earlier. Interstitial lung disease, pretty high if you look at any grade. But when you look at grade 3, it was just about 1 patient. Then the others were fatigue. If you look at fatigue, any grade is nearly about 50%. Nausea, around about 77%, though it is not grade 3, but still it is quite a bit. You had neutropenia and you had anemia, again in the range of somewhere around 30%. And in this study, nearly about 15% of the patient discontinued the drug. We had this Destiny Breast O3 trial, and this has already been discussed by my previous speaker, allowing me to move further. And you are aware of the graphs. This has been discussed just now. And if you look at it, again, the same side effect profile, but this has caused a major revision in the ESMO guidelines, wherein almost TDM1 has been replaced by trastuzumab deruxtecan. Coming to the third one, this is the saxatumab govitecan. It's a murine antibody. It is
Hello,
again,
sir. Sir, kindly conclude your talk in 1 minute, sir.
okay. So again, if we look at it, we go to the data. It was a basket trial. Subsequently, we got 10 milligrams per kg. Results, okay. I wouldn't say after looking at those waterfalls, this doesn't look as good. But if we look at it, safety was okay. Nausea, diarrhea, fatigue, neutropenia, and anemia. In the one case that I had personally used, we had severe febrile neutropenias, so and diarrhea. So coming to it, we come to the trials. We see that the progression-free survival was 5.6 months compared to 1.7 months. We're looking at the drugs on the horizon. I'm really excited about the other drugs also coming in. We have trastuzumab duocarmazine. It's again a HER2 new directed. Dose is 1.2 milligram per kg. We have the TULIP trial. In TULIP trial, it was post-TDM1 versus physician's choice, a median PFS of 7 months, OSHR of 0.83. So probably not as good as the TDX1, but we have other drugs coming up. We have datopotamab deruxtecan. We have something against ladiratuzumab vedotin, again in HER2 negative patients. It has a CR rate of somewhere around 64%. So this is something we have TNBC. Most of the patients were TNBC, and we achieved a clinical response rate of somewhere around 64%. These are some of the drugs that we would be looking at. With this, I conclude my talk, and if there's any questions, I'd be happy to take them. Thank you.
So thank you, Dr. Deepak. So